The overall goal of this experimental protocol is to describe molecular alterations in human diabetic corneas and demonstrate how they can be alleviated by adenoviral gene therapy in organ-cultured corneas.
This method can help answer key questions in the diabetic corneal field, such as what molecular alterations underlie delayed diabetic epithelial wound healing and abnormal expression of epithelial stem cell markers.
The main advantage of this technique is that it allows testing of gene therapy approaches targeting various mediators of corneal epithelial wound healing and stem cell function in organ-cultured corneas.
The implications of this technique extend toward therapy of other corneal diseases related to stem cell dysfunction because we have demonstrated the validity of the method for normalizing altered diabetic corneal stem cells.
Place the corneas epithelial side down in sterile 60 mm dishes and fill the concavity with 0.5 milliliters of agar collagen mixture. The mixture solidifies on the corneas within 2 minutes. Once solidified, place the corneas agar side down in sterile 60 mm dishes containing enough full supplemented medium to reach the level of the limbus for air-liquid interface culture. Then place the dishes containing the corneas into a 5% carbon dioxide incubator humidified with a water pan at 35 degrees Celsius. To perform an epithelial debridement of the central cornea, first soak a 5 mm filter paper disc in N-heptanol and place on the central corneal anterior surface for